Hi and welcome to Onyx Science. This video is about genetics and the monohybrid cross. Here we have three gentlemen, and we might be asking ourselves the question, why are these gentlemen of different heights? Why are two tall and one short? Well, to answer that question, we need to begin by defining three key terms in genetics. The first one is the allele. It's a gene option. So a gene, in this case tallness, might have two different options. You might have the tall version, called the dominant allele, or it might be the short version, which in this case is recessive. So we're going to give the letter A to describe tallness, capital A to describe someone who's tall, because it's the dominant, and then the little a to describe the short, which is the recessive allele. The second term you need to know is the term genotype. And this is the combination of alleles that any diploid organism would have. So we could have big A, which is described as homozygous dominant, homo meaning same. So we have two dominant alleles that are together. We could have the opposite, homozygous recessive, little a, or we could have heterozygous, hetero meaning different. So that could be a combination of a big A and a little a. But look what happens. When we add the genotypes to the individuals, we see that the gentleman on the far end is short with little a, but the gentleman in the middle and the far left both have a single dominant, but the one in the middle is big A, little a, heterozygous. The last term we need to understand is called the phenotype. So if the genotype is what your genetic combination of alleles is, then your phenotype is what does that allele mean when it actually gets expressed, when protein synthesis actually happens, what do you get as a result? So this is called the physical expression, the genotype. And the answer comes down to, well, are these individuals going to be tall or are they going to be short? Well, depending on the combination of alleles, that is their genotype, it defines what their phenotype will be. So in this case, the gentleman that is big A will be tall because he has the dominant gene, or the dominant allele, excuse me. The gentleman in the middle is also tall because he has the dominant allele. And then the gentleman on the far right is short because he does not have a dominant allele. So if we were to redefine dominance, it would simply be the allele that is expressed when it is present. The recessive, then, is the allele that is allowed to show when there is no dominant. But the neat thing is we can start making predictions once we know genotypes. For example, here we've got a couple. She's tall and he's short. What are the probabilities that this pair will produce tall or short children? Well, we can actually predict that, and this is called a Punnett Square, or a monohybrid cross. Here we're going to start by defining the genotypes and determining the genotypes of these individuals, of these parents. So to do that, most text of the question will either tell you what is dominant or what is recessive, and will either tell you what their genotypes are, or ask you to figure them out using other information. In this case, we know that we can see that she's tall and he's short, and we're going to tell you that she's going to have a homozygous dominant trait, and he's going to be homozygous recessive, little a. Number two, we're going to draw up the Punnett Square. This is an expression of all possible combinations when these two reproduce. So we're going to begin by separating the alleles of their genotype into separate gametes, into sex cells. So this gentleman here we see has a diploid condition of little a. That is, his genotype is homozygous recessive. So when he produces sperm cells, each sperm cell can only carry a little a. There's no other option. The female is big A, that is homozygous dominant. So when she produces egg cells, each egg cell can only carry one of those options, and in this case, each egg cell is carrying a big A. So when we combine those sex cells together to identify all possible combinations that fertilization can provide, here we see that all options combine together to make big A, little a, meaning the female can only donate the big A gene to, or big A allele to her offspring, and the male can only donate the little a. So every single child here is going to give us a big A, little a. So when we examine the Punnett square, we can determine the genotype and the phenotype possibilities. So in this case, the genotype is 100% of all offspring, every single one of their children, whether they have 1 or 10, is going to have a heterozygous genotype that is big A, little a for the tallness trait. Their phenotypes, however, what are they going to look like when I see them? They're all going to be tall. And they're going to be as tall as their mother, or somewhere along that line, but not like their father. And we can make these predictions based on what we understand about genetics. Well, that was kind of a silly example. Tallness actually is far more complex than that. But a more realistic example would be something like the gene for fur texture in dogs. Of the gene, we're going to use the letter B. There are two options. There's wire hair, which is a rough, coarse hair, and this is a dominant trait. Or there's a smooth hair, but a recessive version. So that's little b. So the text here tells us that we have two dogs. One parent is homozygous, wire-haired, and the other is smooth hair. Well, wait, what does that mean? Homozygous means it has two of the same allele. And it's wire-haired, so it's big B. So that parent must be big B, big B. The other dog is smooth hair. Well, to get smooth hair, you have to have two recessive alleles. So in this case, it's little b, little b. So what are the possible combinations that this pair will produce puppies of one or another type of genotype? So we do exactly the same thing. We separate the genotype into gametes, and then combine those gametes in fertilization. And then we just examine all possible combinations. And in this case, we see, just like the couple we looked at in the last example, all possibilities will be big B, little b. Now, there's a couple definitions that are worth examining. The parents, the two dogs at the top, are defined as the parents, and we're going to call them the P generation. Their children are going to be referred to as the F1 generation, or filial generation. So, in the F1 generation, what are the possible genotypes? And here again we see, just like the last example, 100% of the offspring will be heterozygous, meaning when I look at them, their phenotype will have the dominant trait, the wire hair trait, and 100% of them should express that. Okay, let's try another example. We're still working with the fur texture gene for dogs, and we're still looking at two possibilities. There's the dominant wire hair and this recessive smooth hair, but this time both parents are heterozygous. Hetero means that they have the different alleles combined. So each one is big B, little b. So when you combine those together, what do you think will happen? Go ahead, finish off the rest of this diagram, complete the Punnett square, and try to determine what the genotype and phenotype ratios are. So go ahead and pause the video and complete this. Okay, I'm going to assume that you've had a chance to finish this. So let's see what you came up with. If you take the dog on the right and split up the alleles into separate sex cells and sperm cells, and do the same thing for the dog on the left for egg cells, and then explore all possible fertilization events, you see that we get big B, big B, big B, little b twice, and then little b. So what does that mean? It means then that for two heterozygous parents, we're going to get 25% of the time big B shows up. So that's 1 out of every 4 offspring. Heterozygous, meaning big B, little b, shows up 2 out of 4 times, so that's 50%. And then the homozygous recessive, little b, shows up 25% of the time. The phenotype ratio, then, is how often will I see the wire hair trait? Well, anytime you see a characteristic with the big B, then we know it's going to have wire hair. So that shows up 3 out of 4 times, that's 75%, and the 25% of the time are going to be smooth. So we can predict, even if these dogs have only one offspring, that 75% probability that these dogs will have a wire hair offspring. Or if they had 10, we could expect that 7 or 8 of them should have the wire hair trait. So the Punnett Square doesn't guarantee, but it does give us an opportunity to make some predictions. So how about this one? We have 2 wire hair dogs, so they both express the dominant, and as they cross, their offspring is a smooth-haired pup. So let's fill in what we know. We know that they're wire hair, but I don't know what their genotype is. I just know that they have a big B. They also have a homozygous recessive puppy because that's what smooth hair means. So how is that little b showing up in the Punnett Square unless the parents are big B, little b? So if I complete the Punnett Square, I can see all possible combinations. So the question here says, if these dogs reproduce again, what are the chances that they'll produce another smooth-haired pup? Well, smooth-haired pups show up 1 out of every 4 times, or in this case, it's 25%. So I expect that there's a 25% probability that the next puppy will have smooth hair. What are the chances that the puppy will be wire hair? Well, in that case, it's going to be the rest of them, 75%. Another type of question you might come across in genetics is, what is the genotype of an individual? So here we see a dog. It says we have a dominant phenotype, which is wire hair. So that should be easy, right? What's the genotype? It should be big B, big B. Or wait, is it really? I mean, it could be big B, little b. The truth is we don't really know. So which one is it going to be? How could we even find out? And the answer is, we're going to do a test cross. We can take this individual and cross it with an individual whose genotype we already know. In this case, it would be a homozygous recessive or a smooth-haired dog. Let's assume that this dog is big B, homozygous dominant. If that were the case, then all offspring are going to have wire hair 100% of the time, no exceptions. But what if instead we find one of the offspring has the smooth hair trait? Well, the only way that could take place is if the dog in question had a little b allele, and therefore has the genotype of big B, little b. You see, we can use what we know about genetics and probabilities and looking at the offspring from a Punnett Square and work backwards to determine the genotype of an individual. So using what you know about genetics now, respond to the following 2 questions. Make sure you can draw the Punnett Square and explain your claim. I hope this has been helpful.